So this lecture covers SARS-CoV-2 and the disease it causes, COVID-19. So what is COVID-19? Obviously, by now, everybody knows what COVID-19 is. It's a respiratory infection caused by the 2019 novel coronavirus SARS-CoV-2. The word corona comes from the Greek word for crown, and you can see that here, these prominent spike proteins surrounding the circular virus particle that gives it a corona or crown-like appearance. It was identified in 2019 in Wuhan, China, and spread globally fairly quickly. This slide overviews coronavirus taxonomy, and I'll start here at the genus level. There are 4 different genera of coronavirus, and alpha, beta, gamma, and delta are their specific designations. SARS-CoV-2 falls into the beta coronavirus genus, along with HKU1 and OC43. These are common cold-causing coronaviruses, and then SARS-CoV-1, which caused the original SARS outbreak in the early 2000s, as well as the MERS virus, Middle Eastern Respiratory Syndrome virus, the deadlier relative of SARS-CoV-1 and SARS-CoV-2. The alpha coronaviruses are a little bit divergent genetically, and they also recognize a different receptor. So 229E and NL63 are also common cold-causing coronaviruses. This slide gives you a nice overview of the replication strategy for the virus, as well as what receptor it's looking for, in this case ACE2, part of the renin-angiotensin system that regulates blood pressure. The virus also requires a protease on the surface of host cells, in this case TMPRSS2, but also furin protease. So that diversity of proteases that can proteolytically cleave the spike protein to facilitate tight binding and insertion of the virus into the cytoplasm becomes really important. The virus will release its plus-stranded RNA into the cytoplasm of the host. And so here's our plus strand of RNA. The plus is a reference to the fact that this can be directly translated into polyproteins. These polyproteins then have to be processed by a protease that the virus encodes. And so that becomes a really great way to target the virus, actually. So Paxlovid is an example of a drug that targets the virus protease to prevent proteolytic cleavage of the polyprotein into individual functional proteins. The full-length genomes can be transcribed into minus-strand intermediates, which can then be transcribed into full-length genomes that can then be used to insert into a developing virus particle. The full-length genomes also encode the subgenomic RNAs that make up a lot of the structural and accessory proteins. So rather than making a full-length genome for every individual structural protein, they make these subgenomic fragments, and then they are translated into proteins that are trafficked throughout the endomembrane system. So here you see them assembling on the endoplasmic reticulum. They bud off through the Urgic system to the Golgi, where they're glycosylated and so forth, and then bud off from the Golgi. Here you see one in a budding vesicle, and the virus forms actually in these budding vesicles, and that's where the full-length genome is associated with the virus proteins and the membrane to ultimately make a full-length virus particle. The virus particle is then extruded into the extracellular environment, where hopefully it's greeted by a robust immune response. In this case, you see an antigen-presenting cell interacting with the virus, and through that process, releasing cytokines to drag more immune system cells to the fight. In some individuals, particularly those with severe comorbidities, there was an enhanced pro-inflammatory response that added to the collateral damage and increased the likelihood of bad outcomes. This slide shows a little bit more detail on the binding. So here you see the virus, and it actually can recognize sialic acids, which are abundant on host cell surfaces. Influenza also uses the sialic acids as a receptor, but this looser binding gives way to tighter binding on the surface of a host cell through ACE2. And then when ACE2 and one of the proteases, again TMPRSS2 or furin protease, interact, they can cleave the receptors on the virus, allow tight binding, and then ultimately insertion of the plus-stranded RNA. So over here you see a little bit about spread. Now the virus can spread through the canonical pathway, like I mentioned, where the virus is assembled in one of those budding vesicles off of the Golgi, and then it's released into the external environment where an immune response can attack it. But what can also happen is the virus can spread cell to cell through syncytia formation. So this infected cell will display virus spike proteins on its surface, and those can bind to the cognate receptors on a neighboring cell. And when that happens, and the proteases cleave those, you get a membrane fusion event, just like this here, where the virus particle fused with this membrane. In this case, however, it's the neighboring cells fusing together. The result of that is just a little bridge where plus-stranded RNA, full genomes, can move from cell to cell without having to interact directly with the immune response. And that increases the efficiency of virus transmission and spread throughout the body. So the presence of these specific kinds of protease on the surface really enhances that process. And so this will eventually, I'll come back to, but that furin protease that I mentioned facilitates this syncytia formation and enhanced spread. And that gets to a part of the story that I'll talk about on virus origins. So here's another overview. This one's showing a little bit more detail of virus architecture, or ultrastructure, if you will. The N proteins you see here are nucleocapsid proteins that interact with the RNA and protect it from degradation. Here's a envelope protein, the prominent spike protein, and membrane proteins, and here's the RNA genome. Here's that interaction between ACE2 and TMPRSS2 or furin, and there's the insertion event, membrane fusion that you see here, followed by the insertion of the viral RNA. Again, the viral RNA is transcribed into more genomes and translated into more virus parts to ultimately make fully formed virus particles, which interact with the immune response. Here you see a dendritic cell or antigen-presenting cell engulfing the virus and then displaying antigens on its surface. This dendritic cell will, so to speak, shake hands with a T helper cell, with a specific T helper cell with the right T cell receptor to recognize that antigen. This stimulated T helper cell will in turn stimulate cytotoxic T cells to kill intracellularly infected cells, but it will also interact with specific B cells capable of making antibodies, which would then bind to and neutralize virus particles. So the immune response is very complex and relies not just on antibodies, but also these other types of immune responses, such as the cytotoxic T cells. So one of the negative outcomes, obviously, is the so-called cytokine storm and specifically acute respiratory distress syndrome. So the virus particles interact with respiratory epithelium. And so, again, this cytokine storm can happen through infections by a variety of different viruses, not just SARS-CoV-2, but influenza and RSV and hantavirus as well. These cells will produce cytokines and chemokines that as a result of the virus infection, that will attract immune system cells to push their way through capillary endothelium. As they push their way through, they cause a little bit of damage, they cause a little bit of leakage, and that leads to the swelling and drop in blood pressure that might be associated with infection. As more of these cells come to the fight, they produce more and more cytokines, which means more and more capillary leakage and more inflammation in that localized region. And again, this localized inflammation is basically collateral tissue damage that can cause an individual to have difficulty breathing and so forth. Much of this cytokine storm can actually damage these endothelial cells as well as enter the systemic pathway and cause inflammation in the blood. And in particular, blood clots might be one of the more negative sequelae. So this is a little bit more of a detailed look at how we might have problems. And so when you compare a low-risk individual to a high-risk individual, these are healthy NK cells. Natural killer cells are designed to kill intracellularly infected cells, similar to the cytotoxic T cells, but less specific. So these healthy NK cells from a low-risk individual quickly recognize antigens on the surface and mark this for destruction, knowing that it's infected. This healthy NK cell produces a sufficient cytokine response to attract effective immune system cells to the fight. And so any viruses that escape will be greeted with dendritic cells and so forth. And any viruses that are piling up inside the cell will hopefully be destroyed along with the cell itself. And the result is the infection is cleared by this productive response. In high-risk individuals, and here I'm talking about people that are morbidly obese, for example, they frequently have dysfunctional NK cells that are incapable of producing sufficient cytotoxins to kill the cell. Therefore, it takes more of them to continuously come, more of them producing more cytokines, which abnormally stimulates the immune response, causing a tremendous amount of collateral damage, essentially spinning their wheels in the mud and getting deeper and deeper in the mud. Eventually, the NK cells become exhausted and the collateral damage is widespread, and you get these symptoms of acute respiratory distress syndrome.